See if we can get this chapter finished up, and then I'll let you guys go around and study and laugh. All right, so like I was saying, with the adaptive immunity, we're now getting specific, and we're going to create a memory. So when we look at adaptive immunity, the forms of immunity that we have, we're going to say it can either be cellular or humoral. Not like ha-ha, okay, but cellular immunity. This one is termed cell-mediated. Now what that's telling us is we're going to have to destroy the cell directly. When we talk about the cell-mediated, lymphocytes, there we go again, got a whole bunch of those around. They directly attack and destroy the foreign cell or diseased host cell. So it's letting us know that it's getting rid of that pathogen, all right, that is inside the cell. So this is telling us it is intracellular, because the antibodies that are going to be this portion, okay, antibodies cannot do that. Antibodies cannot go in and kill the cell, all right? So cellular means we have to get into the cell directly and kill it. Now, humoral. Humor. What does humor of the body mean? Okay, the fluids. Okay? When they talk about the humors of the body, they're referring to the fluids. So, humoral immunity is one that we term antibody mediated. It is because the antibodies are going to be in the body fluids. All right, so we're not inside the cell now, we are outside the cell. So this one is extracellular, outside the cell. They do not directly destroy the cell. They are going to tag the pathogen. When they tag the pathogen, the body has other mechanisms to destroy it. Now, both of these can be used to attack a pathogen. They just simply have to attack at different stages of the life cycle of a pathogen. So it can be one or the other. It can be a combination of both. Now, when we have the cellular immunity, now, since we have reached specificity, okay, the cell-mediated immunity, I'm going to be using T cells, B cells, macrophages, dendritic cells, most of the cells of my immune system. That means I'm going to have cytotoxic T cells, and I have helper T cells. At this point, the only thing we've mentioned is T cells. But now I'm telling you that I've got one that I label cytotoxic, one I label helper. What does cytotoxic sound like? Toxic to the cell. What about helper T cell?
It helps the cell. It helps the T.
It's going to help something, all right? Now, when I'm at this point for the immune system, where I am now going to have specificity, this means that I've had the involvement of an antigen presenting cell, which means I now have an antigen. Is everybody understanding what the antigen is? What is it?
It's a piece of, takes a piece of the virus and presents it to the,
Let's call it the pathogenic.
or the pathogen, takes a piece of the pathogen and presents it to the T cell.
The antigen is a piece of the pathogen. Okay? So my antigen is a piece of the pathogen. And that pathogen has now triggered my immune response. So, when I look at the cells, in the formation of T cells, like I said, go back and read the formation of T cells, because they are actually pretty cool cells. We're going to say that when we have a mature T cell, that mature T cell is now immunocompetent. Take that term apart. Immunocompetent. If you're competent in something, what does that mean?
You understand it.
You're good to go. All right? So these cells are now good to go for our immune system. They are not going to recognize my own cells as an antigen. All right. Now, the reason for that, okay, T cells have to make sure they're not going to attack my own cells. So what we know is that cells have a protein complex on them. Now, I would not be getting into this except for the fact that this is important. Why is it important to understand that my own cells do not get attacked? When they do, it is clinically significant because I now have an autoimmune disorder. Okay? So, we have these complexes, these major histocompatibility complexes, which is basically some proteins on the surface of the cell. We classify them as to 1 or 2. An MHC1 is occurring on all nucleated cells of my body. Note my terminology. All nucleated cells. Do I have any cells of my body that are not nucleated? Red blood cells. So on my body, I have MHC1s on every cell. Goal T cell, stay away from them. Now, when that antigen presenting cell takes that pathogen and they move it and they present it to the T cell, they're showing a piece of the molecule to the T cell, and that is an MHC2. So if I have that monocyte that has put a piece of that foreign cell onto it, my cell should have my MHC1 saying, hey, whoa, this is my cell. You attack this one. Don't attack this one. Does that make sense? Okay, good. All right. Important for immunity. Now, when this takes place, what we're going to see is that when this begins to attack the cells, the agents, the chemical agents that will be used, we can use the perforants, meaning I take it, I put holes in that that pathogen cell and enter some of those granzymes, and hopefully that cell bursts. That has killed the entire cell, therefore being cell mediated. Does that make sense? Yes, no, maybe? Okay. I can use interferons. That means viruses. Tumor necrosis might be something that has to be a cancer cell that gets killed. We can also, because we are affecting the cell altogether, yes, we want that cell to lyse. We want that cell to burst. It could also be that something, macrophages, the cells come in and they eat the foreign object. It could also be apoptosis, meaning a programmed cell death, meaning that at some point in the life cycle of that cell, it's going to be programmed to burst and die. That's apoptosis. In calling up the T cells, we created a memory of the pathogenic cell. Once we have cleared out that pathogenic cell from the body, the T cells have created a memory of that pathogen. And it's a T cell by way of what we call recall response. Meaning, next time that pathogen, that cell, that pathogen tries to invade us, T cells are going to be called up and you're not even going to get sick. Now, this type of specificity At this point, I have not created antibodies. I have simply used materials that are going to go to the cell, that pathogenic cell, and kill it. That was the goal.